and we are mentored by Dr. Erica Buster. My name is Eileen Sayre, and presenting with me today are Melanie Shapiro and Kelly Gunther. The rest of our team is with us here as well, comprised of Joey Frankel, Lex Matthews, Siddharth Plackett, Peter Wang, Danielle Wilkin, and Lin Hun Chu. For a brief overview of our presentation, we will begin with an introduction to some background material, followed by a discussion of our project and methodology, and conclude with our results and future directions. To begin, type 1 diabetes is an autoimmune disorder seen in 1 in every 400 children and adolescents. In the healthy body, the pancreas participates in the endocrine system in order to help regulate blood sugar metabolism. The beta cells of the pancreas are responsible for producing and secreting insulin into the bloodstream in order to help lower blood glucose levels. Insulin is a powerful hormone that acts to remove blood glucose levels by taking in the glucose in the bloodstream and helping the body either use it or store it for later. However, in type 1 diabetes, the body's immune system targets, attacks, and destroys the beta cells of the pancreas, thus eliminating the body's only source of insulin. Type 1 diabetics, thus, must undergo insulin therapy, which entails multiple daily injections of insulin in order to help give the body the control of their blood glucose that they need. However, this presents multiple challenges, as the body has several complex pathways and mechanisms in order to regulate blood glucose levels, and the body does an amazing job of doing this on its own. And so patients with type 1 diabetes are challenged with the task of approximating when they will need insulin and how much they will need. A common side effect of insulin therapy is hypoglycemia, better known as low blood sugar. Hypoglycemia is a dangerous condition because our brain has a very high metabolic demand for glucose, and in hypoglycemia, these demands are not met. In acute cases of hypoglycemia, impairments to cognition and memory can occur, but in more severe cases of hypoglycemia, permanent brain damage can result with corresponding behavioral and cognitive deficits. The brain is so sensitive to the lack of glucose that it has been shown that even a single moderate episode of hypoglycemia has been shown to cause depressive-like behavior and social anxiety. Our project is interested in understanding how certain brain structures might mediate hypoglycemia's effects on anxiety and depressive-like behavior. Specifically, we focus on hippocampus, a structure named after the observation that it resembles a seahorse. Hippocampus is part of the seahorse's scientific name, and so this brain structure is named so. The hippocampus is located within the medial temporal lobe of the brain, as indicated by the arrow above, and it is bilaterally symmetrical and rests below the outer cortex. So it rests in the middle of the brain, and it is bilaterally symmetrical, and can be thought to resemble two bananas attached to the stem. I'll be visualized that way. In regards to what the hippocampus does, the hippocampus is responsible for two broad domains of functions delineated by spatial region. The dorsal hippocampus is primarily responsible for mediating spatial memory and learning. This is what the hippocampus is best known for. The ventral hippocampus is known for mediating emotional regulation and the neuroendocrine regulation of the stress axis. This has been shown to be true through various studies where researchers will lesion part of the hippocampus and observe behavioral outputs that occur subsequently. When the dorsal hippocampus is lesioned, researchers find that animals such as rats and mice are unable to complete mazes or find their way through a pool of water, indicating that their spatial learning and memory has been eliminated, but their emotional regulation is generally unaffected. Conversely, when the ventral hippocampus is lesioned, researchers find that anxiety-like behaviors decrease, but spatial learning and memory are preserved. Again, our project is specifically interested in understanding how the hippocampus might mediate anxiety and depression. We are interested in how depression affects how the hippocampus mediates depressive-like behavior, because some studies also show that the hippocampus mediates depressive-like behavior. And anxiety and depression tend to be comorbid, which means that in clinical cases, they appear together. And so our research question is as follows. How does a single episode of moderate hypoglycemia affect depressive and anxiety behavior? Now I'll pass it on to Melanie to present our
Thank you, Eileen. First, I'll be going over our experimental timeline to give everyone an idea of exactly how we ran all of our experiments. We began by receiving a set of rats and allowing them to acclimate to our animal colony room for a minimum of 5 days. We then habituated our rats to their rodent restrainer for 5 consecutive days, which they were then placed in the following day in order to monitor blood glucose levels. That day, the animals were either given an episode of moderate hypoglycemia via an injection of insulin, or sham hypoglycemia via an injection of saline to serve as our control. 24 hours later, 2 different behavioral tasks were performed. The first was the elevated plusmies test to assess anxiety-like behavior. This was immediately followed by the 4-stone task to assess depressive-like behavior. Our animals were then confused. We habituated our animals to their rodent restrainer in order to minimize the confounding stress response that they might exhibit by being in this closed environment. Stress can impact almost every single aspect of our experiment. Stress is actually known to cause elevations in blood glucose levels, which would counteract our intended hypoglycemia. Stress can also change behavioral outcomes, which would affect both of our behavioral tasks, and can also make our animals more difficult to work with. So we aimed to eliminate as much of that as we could by exposing our animals to the restrainer for 5 minutes and for 5 days prior to the episode of hypoglycemia. Also at the same time of day that they would then be given that episode of hypoglycemia. In order to induce hypoglycemia, our blood glucose level target was 25 to 50 milligrams per deciliter for 1 hour. This was based off of a previously established protocol in a paper where they had shown 1 episode of moderate hypoglycemia to increase depressive-like behavior, however, they did not assess anxiety-like behavior. For our project, we induced hypoglycemia by taking baseline blood glucose levels using the One-Touch Ultra glucometer, and then removing food so as to aid in lowering of blood sugar. We then either performed an insulin injection to induce hypoglycemia or an injection with saline to serve as our control. We continued to test blood glucose levels every 15 minutes until the animals that were given the insulin injection had reached our blood glucose level target. However, sometimes the blood glucose levels of rats given insulin did not end up lowering to this target due to experimental error, and we performed an additional correction with insulin. On the other hand, sometimes blood glucose levels dropped to our target and then continued to drop into severe hypoglycemia. And in this case, we performed corrections with dextrose, an alternative form of glucose. In order to terminate our hypoglycemic episode, our goal was to attain euglycemia, which we defined as greater than or equal to 100 milligrams per deciliter for 1 hour. We first gave a dextrose injection to the rats that were originally given the insulin injection, and then returned food to all the animals. We continued to test blood glucose levels every 15 minutes. Again, sometimes due to experimental error, blood glucose levels did not return to euglycemia immediately, in which case, if they continued to fall, we performed an additional dosage of dextrose. We then continued to monitor blood glucose levels for every 15 minutes until blood glucose levels were rising or stable. The next day, we performed 2 different behavioral tasks. The first was the elevated plusmies to assess anxiety-like behavior. As you can see, this task involves a setup with 4 arms. 2 of the arms are open and 2 are closed. To begin, an animal is placed into the center of the maze and allowed to move out into any of the arms that it wishes. Its motion is tracked for 5 minutes via the EcoVision video tracking software. Animals that spend more time in the open arms are indicative of having less anxiety-like behavior. On the other hand, animals that spend more time in the closed arms are indicative of having more anxiety-like behavior. Another thing that we look for is for animals that peer over the side of the open arms, which we term the head dip region, also indicative of less anxiety-like behavior. This was then immediately followed by our second behavioral task, the forced swim task. In this task, animals are placed into a pool of water, and again, their motion is tracked for 5 minutes via lethal vision. Animals that spend more time in the mobile phase are indicative of having less depressive-like behavior, while animals that spend more time immobile are indicative of having more depressive-like behavior. Essentially, an animal that is immobile has given up from trying to escape this stressful task and the stressful environment that they were placed in. Now I will pass it on to Kelly to talk about some of our results.
Thank you, Melanie. So our analysis of data in this experiment was twofold. The first step was to ensure that we had a protocol that could successfully induce hypoglycemia to our specifications in our rats. So on the graph here, if I can orient you to the axes, the x-axis is time in minutes, and the y-axis is the average blood glucose level of the rats in milligrams per deciliter. The black line marked with squares represents the rat who received the saline injection, while the gray line marked with triangles represents the rats who received the insulin injection. And so here we are marking the blood glucose level trajectories of the rats who were administered a 0.005 units per gram injection of insulin with their blood glucose levels checked every 30 minutes. And so we found that with this protocol, we were in fact able to induce hypoglycemia. However, the blood glucose levels dipped lower than we had planned. Our goal was for the rats to return to euglycemia at 135 minutes. However, this was not the case. The error bars and asterisks represent that there are significant differences between the average blood glucose levels of the saline rat versus the insulin rat. So from this, we kind of established that measuring blood glucose levels in 30-minute increments was not allowing us to plot their blood glucose level trajectories as accurately as they could, and so we needed to revise our experiment. The problem with plotting in 30-minute increments was when their blood glucose levels dropped below 15 milligrams per deciliter, it made it very difficult for the rats to reestablish euglycemia. You can see here the black bar marks the frequency of rats who were able to reestablish euglycemia, while the gray bar marks those who remained hypoglycemic. So while 2 of these rats could reach levels of severe hypoglycemia, were able to reestablish euglycemia, 5 were not. So this marked a revision in our protocol. So our next step was we decreased the insulin injection to 0.004 units per gram and decided to take blood glucose levels in 15-minute increments rather than 30. And we were successful in inducing moderate hypoglycemia with this protocol. Again, the x-axis is time in minutes, and the y-axis is average blood glucose level in milligrams per deciliter. And so you can see here that at the 0-minute time point, the rats started at levels of euglycemia. By the 45-minute time point, they had reached levels that we defined as moderate hypoglycemia. This continued to the 90-minute time point, and then at 135 minutes, they were able to reattain euglycemia. These brackets and asterisks show that the average blood glucose levels during periods of euglycemia were significantly different than the average blood glucose levels of the times of moderate hypoglycemia. So next, after we induced hypoglycemia successfully, it was time to complete the behavioral tests. As Melanie mentioned, we used the elevated plus maze to assess anxiety-like behavior. So if I can orient you to these pictures, this is an overhead view of the elevated plus maze. The horizontal bars that I've indicated with the red arrow are the closed arms, and then the vertical ones are the open ones. So we have a video of the rats completing the maze. you is that the saline rat takes a visit to the head dip region, where you can see him reaching his nose over the edge of the open arm as if he's exploring his environment. So we used ecovision to more precisely tabulate the amount of time the rat spent in these different delineated regions on the elevated plasmies. So from this point forward, the black bars will represent the rat who received the saline injection, and the gray bars will represent the rat who received the insulin injection. So we found here that the rat who received the saline injection had a higher frequency of visits to the head dip region than the insulin-treated rat. So this is indicative that hypoglycemia increases anxiety-like behavior. We also we tabulated the cumulative duration in seconds that each rat spent in the different areas of interest on the elevated plasmies, this being the closed arms, the center, and the open arms. So we found that the saline-treated rat spent less time in the closed arms than the insulin-treated rat, more time in the center than the insulin-treated rat, and a little bit more time in the open arms than the insulin-treated rat. This is indicative of higher levels of exploratory behavior for the saline-treated rat as compared to the insulin-treated rat, which again would suggest an increase in anxiety-like behavior. So next we have the forced swim tests. And so these videos look approximately the same, which is why it became in great use to have ecovision technology available for us, because it was able to very precisely tabulate the number of periods of immobility versus mobility as the rats completed the test. So we tabulated the number of bouts of immobility that each rat experienced, and we found that the saline rat had less total bouts of immobility than the insulin-treated rat, which would suggest that hypoglycemia increases depressive-like behavior. We also found that the saline-treated rats spent a lower percentage of their total time in the immobile phase as compared to the insulin-treated rat, which again would suggest a higher level of depressive-like behavior. We also calculated the latency for immobility. So how long did it take the rat from the first second it entered the tank to have its first period of immobility? And we did find that the saline-treated rat had a shorter latency for immobility than the insulin-treated rat. This is not necessarily in line with what we would expect if the insulin-treated rat was exhibiting higher levels of depressive-like behavior, because theoretically they would have a shorter latency for immobility. However, this is just what we found in our data collection. So as an overview, our conclusions from the data analysis would be that a 0.004 units per gram dosage of insulin with their blood glucose levels checked in 15-minute increments is the best methodology to successfully induce moderate hypoglycemia in our rats. We found that this episode of moderate hypoglycemia led to an increase in anxiety-like behavior, as well as an increase in depressive-like behavior. Now, we did note some limitations in our methodology as we completed this experiment. You may have noticed that I referred to singular rats throughout the talking about the videos, and that is because we only had an N value of 1 for both the experimental and control group. This was due to difficulty in the inducing hypoglycemia protocol as a whole, as well as in restoring euglycemia in our rats. Also, we recognized that stress from the restraint system, as well as the testing environment as a whole, could have impacted behavioral results. While we took every step necessary to habituate the rats to both their colony room as well as to the restrainer environment, it was still a new and potentially stressful experience for them, and this could have really changed the way they behaved in either of our behavior tasks. So I'm going to pass it back to Melanie to discuss clinical relevance and future directions.
Thank you, Kelly. So as our study was performed with animals, it's important to note how this can be applicable to humans, and also how this can be translated potentially to clinical studies. A number of clinical studies have found that not only does hypoglycemia itself lead to anxiety and depression, but that many diabetics have anxiety and depression due to a fear of developing hypoglycemia. So this preceding anxiety and depression could confound any anxiety and depression found after the episode in terms of being able to discern which of these behaviors are actually due to the physical episode of hypoglycemia. Additionally, the study has shown that glycemic variability, so hypo and hyperglycemia, both increase anxiety and depression. Overall, an awareness of mood changes is very relevant to both diabetics and their caretakers. So, to summarize, even though there are many variables that make this very difficult to study in humans, it's important to be able to use animal models to more tightly control blood glucose levels in these types of studies. Here are some of our future directions. We would like to improve our protocol for reducing hypoglycemia. This could be done via a hypoglycemic clamp, in which dextrose and insulin are co-infused in small amounts continuously. This aids in greater control of blood glucose levels. Additionally, we noted that another paper used an alternative administration method for injecting dextrose. Actually, instead of an injection, they used oral lavage, in which dextrose would be administered directly to the stomach. We also would like to analyze new cell birth in the hippocampus in order to be able to correlate some of our behavioral changes with changes within the brain. This could be done with staining for bromodeoxyuridine, also called BRDU. Bromodeoxyuridine is an analog for thymine, and as such, it can become incorporated into the DNA of newly proliferated cells. We would expect with this type of experiment to see new cell birth in the hippocampus to decrease in insulin-treated animals as opposed to the saline-treated controls, and that this decrease could be correlated with an increase in anxiety and depressive-like behaviors. We would like to acknowledge a lot of people. First of all, our mentor, Dr. Glasper, her graduate students, Molly and Shannon, who helped us along the way, the Gemstone staff for all of their support, our librarian, Dr. Guy Pacheca, the Howard Hughes Medical Institute for their generous funding, and all of our discussants for attending today and for reading our thesis. Thanks so much.
Like I promised, we're going to start with about 10 minutes of questions restricted to our discussants to get them warmed up for what's going to come later when we kick them in the back room. So what I'd like the discussants to do is when you offer your question, please give me a very brief introduction to who you are and why are you a discussant for this group, what experience you and how you plug in, if you will, and then pose your question then. And we discovered that you need a microphone. So I'll be running the microphone around as needed, and when it gets time for audience questions, I'll run it to you too, or else we won't hear anything. Most people can't fill the room. Like some of us. Who would like to go first?
My name is Jessica Shokoko. I'm a consultant here, I would say, because I teach general endocrinology. So I'm more interested in the insulin aspect of it. And my question is, what giving insulin result in hypoglycemia that can affect the brain function, right? Because you can give insulin. What is happening that will affect brain function?
Okay, so the question was, how does insulin affect brain functioning if it causes hypoglycemia? So when insulin arrives in the bloodstream, it serves to remove it from the bloodstream so that the body can either use it or store it for later in the form of glycogen. The brain, again, has a really high metabolic demand for sugar. It weighs a very small amount of our body weight, but needs around 20% of the glucose and oxygen that we use. And so glucose is transported into the brain, it crosses the blood barrier region, but when hypoglycemia sets in, this glucose is not delivered. And so the brain must resort to alternative sources of energy. So some of them include lactate, for instance. This is transported into the brain with MCT transporters, for instance. But in regards to how the delivery of insulin affects brain function, I guess, when glucose is not able to be delivered into the brain, neurons don't have that source of energy, I guess, to engage in the metabolic processes to fuel proper firing of the neurons and such. So. The question was, are neurons insulin-dependent cells? If I remember from your class correctly, I would hazard the answer that they are not insulin-dependent. Nonetheless, they still require, well, okay, I see where you're coming from. We still see that the lack of glucose is correlated with the behavioral effects of impaired judgment and memory. And I think we experience that behaviorally and anecdotally on our own. We know that when our blood sugars are low, we have trouble focusing and things like that. So.
Great. Next question we're discussing. Yes.
Hi, Matt Resch, I'm faculty in the psychology department. So I guess kind of a follow-up question to that. So it's going to impact the entire brain, right? So, but you seem to focus on the hippocampus. I wonder if you could talk about other brain areas that might be involved.
So we focused on the hippocampus because we found in papers that that was the area of the brain that was most susceptible to hypoglycemia-related damage. However, there were other papers to show that the cerebral cortex was also somewhat vulnerable, as well as I believe the basal ganglia. And so there have been many papers that have looked at neuronal death as well as different behavioral tasks. But because the hippocampus was the most susceptible, that appeared to be the best choice for us.
Why is it?
Oh, sorry. The question is, why is the hippocampus more sensitive? I think that that is something that researchers aren't entirely sure of. They have only found in, I think, one paper that the hippocampus was more susceptible. So I think that within that paper, they weren't entirely sure of the reason why. They hadn't actually searched for the mechanism. And that paper, there may have been others.
Okay. Next is Lee.
Richard Yee, Professor Oliver. So, let's say you got great data, a bunch of organizations, consistent with your predictions. What is the clinical implication of those results? And more importantly, what would be the limitations in how you interpret those results given the specific design that you implemented?
All right, Doctor, you want to pick somebody? Kelly's got it.
Okay, so there was a lot of literature that highlighted hypoglycemia in terms of anxiety. And so that was kind of a natural choice that it increased it. Well, we weren't sure. We knew we would see a change in anxiety-like behavior, but we weren't exactly sure what the directionality of that would be. And unfortunately, we didn't get the significance in our data to make a decision one way or the other. We were interested in depression due to its high comorbidity with anxiety. While researchers do, there's not really a very clear point to depression. It can also be associated with the hippocampus. There's the neurogenesis theory of depression, which suggests that decreased neurogenesis can lead to an increase in depressive-like behavior. So the clinical implications of this would be just better understanding the cognitive risks that type 1 diabetics might face if they do who have moderate or severe episodes of hypoglycemia. We examined moderate, but severe, I think, would highlight an even larger body of problems that could happen just from the deprivation of fuel to areas of the brain.
And we have another discussion question? Okay.
Hippocampus is neat only looks. Actually, in the brain, it's the only region that you actually replace with stem cells, which means the metabolic requirements will be very high. But my other question is, what about neurotransmitters? Is the hypoglycemia affecting a specific neurotransmitter that is mediating the aggressive episode? Because we know that the hippocampus is part of the limbic system, so there should be opioid receptors somewhere. So, can you talk about something like maybe it's reducing, say, endorphins? Any other nutrients, any other neurotransmitter?
I'll walk slowly. Who wants to come up? We might have to let that go for the further discussion in the back room. I think between now and then they'll get together and huddle and come up with a pack plan. That's not uncommon when you have questions thrown at you cold. That's what you have. Think about it, come back later. Okay.